Hello viewers, today we will solve paper 2 biology 5090 May-June 2019. So let's we start from question number 1. Question 1 is, food supplements are substances that are often added to a person's diet. These supplements may be in the form of a tablet. The diagram shows the label from a food supplement to be taken with a balanced diet. The components are amylase, cellulase, lipase, protease. So different types of enzymes have been given. This supplement may be taken by a person whose pancreas is not producing enough or some of the components shown on the label. State the type of chemical that the supplement contains. Okay, in the table, 4 different types of enzymes are given. So all enzymes are made up of protein. So when we talk about the state the type of chemical that the supplement contains, actually all enzymes are made up of protein. So the right answer for this question is protein, or we may write enzymes. Then move to part 2, question 1. Each food group in the diet is a substrate for a specific component of the supplement. Complete the table by writing the most appropriate word or words in each box. In this question, we have been given the components of supplement: amylase, protease, and then lipase. The substrate, these enzymes will be used to catalyze the reaction and to convert them into the final products. So let's see what will be the answer over here. When we talk about the substrate, first component of supplement is amylase. So amylase always catalyze the substrate, that is starch. So in our answer we will write starch over here. So let's we write starch. The answer is starch. Second, the component of supplement is protease. So the answer over here, protease always digest protein. So over here we will write the answer protein. The third component is lipase. Lipase digests fats or lipids. These are the substrates. We will talk about the final components, the final components we get after the reaction. Mean when amylase digests starch, this amylase will convert this starch into maltose or glucose will be the final product. Maltose or glucose, that's the final answer. When we go with protease to digest protein, protease will digest this protein into polypeptide or amino acids. polypeptide or amino acids. Finally, lipase will digest fat into fatty acids and glycerols. Fatty acids and plus glycerols. These are the final products. Okay, now move to next question. Next question is, cellulase is a component of the supplement that is not usually found in the human body. Use your knowledge of the structure of a plant cell to suggest the function of the cellulase component of the supplement. Cellulase is enzyme which digests cellulose. In human beings, cellulase is not produced, so we cannot digest cellulose. In this question, the examiner is going to ask you to use your knowledge of the structure of a plant cell to suggest the function of the cellulase component of the supplements. So now we move to answer this question. Let's we answer this question. Cellulase, as we told you earlier, is an enzyme which digests or which break down cellulose. Cellulose is broken down by cellulase enzyme. So this is the main function of this cellulase enzyme, to break down cellulose, to digest cellulose. Move further. Part B. The supplement is taken in the form of a tablet that is swallowed. Each tablet is covered in a substance that breaks down only in alkaline conditions to release the components of the supplement. Suggest why this covering is important if the supplement is to work effectively in the alimentary canal. In our alimentary canal, two places are very dominant where the process of digestion takes place other than the mouth. The first one is stomach and second is small intestine. In this question, the examiner is asking, suggest why this covering is important if the supplement is to work effectively in the alimentary canal. In what the covering is actually, the covering is substance that breaks down only in alkaline solution. It means nothing will be going to happen with this tablet, but it will be in stomach because it is acid resistant, and it only will be broken down when it will be inside small intestine where the conditions are alkaline, particularly in duodenum. So when we answer this question, what we have to write? Let's see. First we draw the lines. 3 points we have to give. Actually there are 3 marks, so keep in your mind at least 3 marks must be given against this question. So when you start writing the answer, keep in your mind your stomach is having acidic conditions, or the conditions in your stomach are acidic. So your tablet does not or will not be broken down in acidic conditions. So start writing your answer. Conditions of stomach are acidic. That is, having pH around 2. When highly acidic conditions are there in your stomach. So in these conditions, tablet will not be broken down. So tablet will not be digested, or it will not be broken down in the stomach. What happens next? It means it will pass from the stomach into the small intestine, into the duodenum. When it reaches to duodenum, where pH is above 7, where pH is above 7, tablet will be broken down tablet will be broken down to release contents. So this is the answer you have to write against this question. Actually, you have to give at least 3 points, as we have given the conditions of stomach where the conditions are acidic. Second, we are explaining the conditions of duodenum. So no digestion will take place, or there will be nothing happen to the tablet in the stomach, because conditions are not suitable for its digestion. It will pass to the small intestine from the stomach, where the conditions are more alkaline, pH is above 7. Over there, this tablet will be broken down and it will release its contents. So this is the answer that you have to write. Okay, move to next question. But before that, what is given in question number 2? In question 2, the diagram shows the structure of a type of bacterium and details of the response of the bacterium to light. Before moving forward, I suggest or strongly recommend to the students, whenever you are giving your answers, particularly against any diagram, try to focus on their labels, what have been given, because always the answer is related to the label. So over here in this diagram, on left side of the diagram, direction of light is given. When light is given, definitely it will be having something with photosynthesis. The first answer that will come in our mind. But let's see what is given in other labels. Second, fibers that attach to a surface and then contract to enable the bacterium to move. locomotion. If flagellum have been given there, definitely the animal either is moving towards the light or away from the light. From second label, if we go into detail, third, light receptors. Again, light receptors are given here, and definitely it is linked either with the images, mean how to capture image, or otherwise mean definitely uh, it is photosynthesis. The process is directly involved in the food production. The last one is structures containing chemicals similar to chlorophyll. Mean again, the last one is giving clear cut image. Mean this light is there for the process of photosynthesis. Okay, now we move to question. Used information in the diagram to suggest the sequence of events that takes place when the bacterium is provided with light from the direction shown. In this question, actually there is a relationship between the bacterium and the light. So means you need not to discuss anything regarding photosynthesis, because over here you just have to give the reaction of the bacterium against the stimulus. Light is stimulus, and we have to answer whether the bacteria is moving towards the light or away from the light. Actually, when we link it with chlorophyll, then definitely it will be moving towards the light. So what we have to write in our answer? Let's we move to answer the question. So actually again 3 marks are given against this question. So let's see what we have to write over here. In this question, I mean definitely first we will focus the light. When this light converged and travels to the bacterium, so how we will write? Light after converging travels to bacterium. Mean this light will reach to bacterium. Then what happens? Sensory cells are given, or receptors are given on the body of bacterium. Actually these sensory cells or receptors are there to absorb or to detect light. So next we will write light after converging travels to bacterium, and receptors what receptors will do? These receptors will detect the light. Receptors will detect light. So when light is detected or absorbed by the bacteria, there will be a movement of fibers. Fibers will contract as a response. As a response, flagellum or fibers contract, and resultantly bacterium will move towards light, and resultantly bacterium will move towards light. So this is the answer that you will write for this question. Move to next section. Let's see what is the next question or next part of this question. In part 2, the advantage to the bacterium of responding to light in this way. So here we will link the advantage of light. First, the bacteria will detect the light and try to move towards the sunlight. When definitely it will be closer to the sunlight, capturing more light. This light will help the bacteria for the process of photosynthesis, because bacteria contains the pigments, chlorophyll pigments in its body. So that's why we will link it with the process of photosynthesis. So 2 marks are given. So let's see what we have to write over here. This question, the answer should be like bacteria will absorb more light, will be absorbed by chlorophyll in bacterium, which will help in the process of photosynthesis to help the process of photosynthesis to produce glucose, starch or carbohydrates. To produce carbohydrates. This is the answer that we have to write in this section. Now we move to next part. In part B, there are similarities between the functions of parts of the bacterium and the functions of structure found in the human eye. Suggest which structure in the human eye has a function similar to each of the following regions of the bacterium. Actually, when we talk about the structure of human eye, we have receptors at retina. We have rods and cones. They are the sensory cells. Actually, they absorb light and give us image. So now we have to link it with those receptor cells present in the eye of human beings. So let's we answer this question. Okay, in part A, we will link it with cornea, because in human eye, cornea is a part from where the light will converge. It will get enter into the eye, it will be converged, and finally it will pass through pupil. So when we link it with A, over here we will write the cornea. A Apart from cornea, we may write lens or any other part through which light will pass. But better to write cornea because it comes first at start of when light will get enter into the eye. So for B, we will write the photoreceptors, mean here we will write the membrane on which these photoreceptors are present. They are the retina. That membrane is called retina, on which rods and cones are present. These are the sensory cells which absorbs light and gives or help us to make an image. So these are the two parts which are connected to this question. Okay, now we move to next question. Or next part of this question, the C. The type of bacterium shown in the diagram fixes nitrogen. Describe the role of bacteria in nitrogen fixation. So this question is actually a part, or it is linked with another chapter that is ecology, ecosystem, and linked with nitrogen cycle. When we study nitrogen cycle, in nitrogen cycle there is a process of nitrogen fixation and there is a process that is called biological fixation. So keeping in mind that biological fixation, we have to answer this question. So let's see what we will write in this answer. When we start writing the answer, we will link it with nitrogen cycle. Before that, let me explain you the process of nitrogen fixation for more clarity. When you talk about the process of biological fixation, over there these are the root hairs which are present in the soil. This is stem. This part is basically a stem. Okay. The upper part where leaves are present, where the process of photosynthesis will take place. Over here, from air, gaseous exchange is going to be taken place, then it is linked directly to the process when we reach to the root hair structures. Over there, node like structures are present here. These are root nodules. In these nodes or nodules, nitrogen fixing bacteria are present. Rhizobium bacteria. These rhizobium bacteria are present in the root nodules. What actually they do? They take nitrogen from the atmosphere. They will take nitrogen from the atmosphere and convert it into ammonia or nitrates. In over here, ammonia or nitrates are made. So this was the process where nitrogen fixation will take place. So we have to link the answer with this concept. These are the bacteria which are actually involved in the process of nitrogen fixation. These bacteria will use nitrogen. So what we have to write in our answer, let's we see. In our answer over here, we will write these bacteria use atmospheric nitrogen to produce ammonia or nitrates. Mean they are fixing nitrogen or fixing nitrogen biologically. So this will be the answer of this question. Okay, now we move to next section of this question. What is that? Let's see. Move to section D. The enzyme that these bacteria use to fix nitrogen is destroyed in the presence of oxygen. Suggest why destruction of this enzyme is likely to occur in the type of bacterium in the diagram. Okay. Actually, here what we have to answer, it's very simple. These are the nitrogen fixing bacteria. Actually, they are destroyed in the presence of oxygen. When there will be more availability of oxygen in that environment, these bacteria will not be functional. They will not be able to fix nitrogen. They cannot work in the presence of oxygen. So how they will get destroyed, right? So let's we see how we have to answer this question for 2 marks. Okay, let's first we draw the lines. Let's write our answer. Okay, in this case, actually, when the process of photosynthesis will take, actually, in the process of photosynthesis, oxygen will be produced. So when this oxygen will be released, obviously it will destroy the bacteria. So keep in your mind during the process of photosynthesis in these bacteria, oxygen is produced or released. So this oxygen will destroy. This oxygen, let's see what he's asking actually. The enzyme these bacteria use to fix nitrogen is destroyed. So it means this oxygen will destroy the enzymes. So this oxygen will destroy the enzymes So when these enzymes are destroyed or denatured, so they cannot carry out the process of nitrogen fixation. So this is the answer that you have to write in this section. Now move to next part. That is the last part of this question. Suggest one adaptation that such bacteria may have developed to prevent the destruction of this enzyme. So actually, whenever you are asked to suggest, multiple answers can be given, because there is no hard and fast rule, no one fixed answer will be there. So many answers, but must be relevant to this question. So keep in your mind, for 1 mark when you will be giving the answer, so what options or what suggestions should be there. And obviously, over here less oxygen should be present, so any suggested method can be used over here, in which we can write our answer. Mean there may be a mutation, definitely mutation will change the behavior of the enzymes, so they may work even in the presence of oxygen, or if lesser amount of oxygen is present over there. So we may write number 1, any method if we can reduce the amount of oxygen. Oxygen percentage or oxygen presence may be reduced. We may reduce the presence of oxygen in that environment by using any method, or we may keep away the oxygen from enzymes. Try to keep away oxygen from enzymes, and we may use mutated enzymes, or mutation can change these enzymes. So mutation is another process, is another option which we may write in this answer. So these are the 3 suggestions or possibilities which we can write in this answer. So viewers, inshallah we will proceed further in next video. Until then, stay safe and Khuda Hafiz.